Okay, good morning to all. My audio and video are clear. Please respond. Audio and video are clear to everybody. Yes. Okay. Most of them are not yet joined, I think, because the timings are changed. Okay. Sleeping. Yes, most of them are sleeping. So when classes begin, regular classes, your class starts by 6:50 here. Okay. So therefore, you have to habituate the timings. Times are very, very important. Okay. Right. So let us continue. Let us start. In the last class, we are discussed about glycolysis, right? So in the last class, glycolysis is completed. What is the site of glycolysis? Where glycolysis takes place in the cells? You know, glycolysis is the first step of respiration, that is both aerobic respiration and anaerobic respiration, correct? Yes. So the site of respiration is cytoplasm. Good. Who discovered glycolysis? Who discovered the scheme of glycolysis? I told you 3 scientists. Okay? 3 scientists, namely Embedon, Meyerhoff, and Parnas. Correct? For their honor, by taking the first letter of their names, combinedly called as EMP pathway, correct? EMP pathway. Right. Okay. The term glycolysis was derived from which language? Is it English language or Latin language or Greek language? Yes. It has originated from Greek. Good. So we are recollecting the concept of glycolysis, and it is essential to understand the next one. That is why. Okay. And glycolysis is the common step for both aerobic and anaerobic respiration. Yes. Is it independent of oxygen or dependent of oxygen? Is it independent or dependent? Glycolysis. Yes, glycolysis is independent of oxygen. That means whether oxygen present or absent, glycolysis takes place. That is why it occurs in all living cells of all organisms, whether it is prokaryote or eukaryote. Understood? Okay. What is the substrate for glycolysis in plants? What is the substrate for glycolysis in plants? Glucose or fructose, correct? Yes, glucose or fructose. Right. These substances in plants are formed from which substance? Glucose and fructose are derived from which substances in plants? Yes, sucrose, which is the end product of photosynthesis, or the stored product, stored carbohydrate, that is starch. Yes. What is sucrose? Is it monosaccharide or disaccharide? Sucrose is a disaccharide, right? Yes, very good. Sucrose is a disaccharide. And this sucrose split into two monosaccharides, namely glucose and fructose. Sucrose is a disaccharide. Glucose and fructose are monosaccharides. And this glucose or fructose enters into glycolysis. Okay. Next, which enzyme is involved in splitting of sucrose into glucose and fructose? Which enzyme? Namely, invertase. Sucrose break down into glucose plus fructose in the presence of an enzyme called invertase. Correct? Yes. Next. Glycolysis consists of how many steps? How many metabolic reactions are present in glycolysis? Very good. 10. 10 reactions. Is it enzyme controlled process? Is glycolysis enzyme controlled process? Yes. All 10 steps are controlled by one enzyme, or each step is controlled by one separate enzyme? Understand my point? Is all the steps are controlled by only one enzyme, single enzyme, or each step is catalyzed by one separate enzyme? Yes, you are right. Each reaction is catalyzed by one enzyme. Therefore, 10 enzymes are involved in glycolysis process. Yes. Okay, now I will ask very important point. What is the first enzyme of glycolysis? What is the first enzyme of glycolysis? Very good. Not all are involved, only few members are involved. You should be active. The last evening only we completed. You have to remember the process. Okay, the first enzyme of glycolysis is hexokinase. The first enzyme of glycolysis is hexokinase. What is the substrate for hexokinase? What is the substrate for hexokinase? And what is the product? What is the substrate and what is the product for this enzyme, that is hexokinase? Yes, substrate is glucose. Yes, product is glucose 6 phosphate. Correct? Substrate is glucose, product is glucose 6 phosphate. Okay, very good, very good. Next, if you are telling the answers, giving the answers, the input is correctly giving, indicates that we are following the subject, you are understanding the subject properly, and definitely this will help you a lot in future also to learn all the chapters, to get clarity in the concept, and ultimately you will get more marks and ultimately you will get the good rank as well. Understand? Right. Because these are basics only we are teaching. We are not in depth till. So definitely everybody should understood clearly. Right? Then in glycolysis, is-it oxidation takes place? In glycolysis, is-it oxidation step is present or not? Yes, yes, very good. Oxidation step is present in glycolysis. Oxidation step is present in glycolysis. Which step is the oxidation step among the among the 10 reactions? Among the 10 steps in glycolysis, which is the oxidation step? Sixth one. Sixth reaction is the oxidation step. How many oxidations occur in glycolysis? How many oxidation? Remember, oxidations occur in respiration. All oxidations are dehydrogenation reactions. All oxidations are dehydrogenation reactions. So only one oxidation occurs in glycolysis. Can you say what is the product of oxidation of step of glycolysis? What is the product of oxidation step? End result, yeah, I'm talking about the product is NADH. NADH is formed during oxidation. What is NADH? I told you it is a coenzyme. Can you say the full form of NADH? Nicotinamide. Adenine dinucleotide. This is the full form. Nicotinamide adenine dinucleotide. That is the product of oxidation. Okay, good. Next. Is it ATP are used in glycolysis? Is it ATP are used in glycolysis? Yes. Yes. At how many steps ATP are used and what are they? At how many steps ATP are used and what are those steps? Very good. Most of them are giving the correct answers, but still some of them are not giving correct answers. I am asking about at how many steps ATP is used. Only 2 steps. That is, first step and third step. In the first step, 1 ATP is used. In the second step, 1 ATP is used. So, ATP are used at 2 steps, first and third. Total number of ATP used are 2. Very good. Okay. Next. Is it ATP is formed at any place? Is ATP are formed in glycolysis? Previously, just now I told you that ATP is used. Is ATP is synthesized or formed in glycolysis? Is ATP is formed in glycolysis? Yes. Yes. In how many steps ATP are formed? 2 steps. Can you say what are those steps? ATP is formed in 2 steps. ATP is formed in 2 steps. What are those 2? 7th step and 10th step. In 7th step and 10th step. Okay. In 2 steps used, in 2 steps formed. But the number of ATP used, number of ATP synthesized are different. Correct or not? Number of ATP used are 2. Number of ATP formed in each step are 2. That means here 2 ATP formed. Here 2 ATP formed. So these are formed. Total number of ATP formed 4 ATP. I told you once one name for this. I told you one name for this. If ATP is directly synthesized during the conversion of one substrate into another substrate, not through electron transport system, I have given one name. Anybody can tell that name? That is a very important, crucial step. Technically, I told you one name. Very good. Kirti. Only Kirti and also some other members. Yes. Yuvraj, Jahnavi Reddy and all these. Not all, very few members are telling SLP reaction. SLP step. SLP means what? Substrate level phosphorylation. Substrate level phosphorylation. I think most of them didn't concentrate on this name. This is very important. SLP means substrate level phosphorylation. So, how many steps? 2 steps. How many SLPs? 4 SLPs. For each SLP, 1 ATP is formed. Do you agree? Total number of steps are 2. Number of SLPs are 4. So that means total number of ATP formed is equal to 4. So how many consumed? How many consumed? How many used? 2. How many formed? 4. Now can you say right now, what is the net gain of ATP in glycolysis? What is the net gain of ATP? Many a times asked this question in competitive exams like NEET. Net gain of ATP in glycolysis is equal to 2. Totally formed 4, used 2, so net gain will be 2. Very, very, very important. Okay. Right. Now, is at any place in glycolysis oxygen utilized? Is at any place oxygen used? I told you initially that glycolysis is independent of oxygen, correct? Glycolysis is independent of oxygen. Yes. So oxygen is not utilized. Is at any place CO2 released? Is at any place CO2 released? No. No. Good. So CO2 is not released, oxygen is not consumed. Right. Now, what is the end product of glycolysis? What is the end product of glycolysis? What is the end product of glycolysis? Pyruvic acid. The end product of glycolysis is pyruvic acid. Can you say, anybody, how many carbons are there in pyruvic acid? How many carbons? Very good. It is a 3-carbon compound. It is a 3-carbon compound because one molecule of glucose break down into 2 molecules of 3-carbon pyruvic acid. Two molecules of 3-carbon pyruvic acid. So this is the end product. Okay. Now a question may be asked like this also. What is the key product of glycolysis? What is the key product of glycolysis? Pyruvic acid. Pyruvic acid is the key product of glycolysis. The metabolic fate of this pyruvic acid depends upon cellular need. Now we enter into the next concept. The metabolic fate of this pyruvic acid depends upon cellular need. That means the fate different if oxygen is present. The fate of pyruvic acid is different if oxygen is absent. That means presence or absence of oxygen decide the fate of the pyruvic acid. Because in all the cells, pyruvic acid is the end product. At the end of glycolysis, pyruvic acid is the end product. Whether it is aerobic organism, anaerobic organism, the first step, the common step is the glycolysis. So glucose or fructose ultimately break down into pyruvic acid. Now the pyruvic acid fate is decided by the presence or absence of oxygen. Now let us see. This is the pyruvic acid, a 3-carbon acid. The fate of this depends on oxygen availability. Okay, let us see now. In the presence of oxygen, it is converted into acetyl coenzyme A, and ultimately it is converted into carbon dioxide plus water. This is called aerobic respiration. I think, understand. If oxygen is available for this pyruvic acid, the pyruvic acid first converted into acetyl coenzyme A, then the acetyl coenzyme A through number of steps ultimately break down into carbon dioxide and water. This is what is called aerobic respiration. All this happens in the presence of oxygen. If no oxygen, if it is yeast is a fungus, unicellular fungus, yeast is a unicellular fungi. In the absence of oxygen, no oxygen, in the yeast cells, this pyruvic acid is converted into ethyl alcohol plus carbon dioxide. This is called alcohol fermentation. Alcohol fermentation. If it is a bacteria, the pyruvic acid is converted into lactic acid. The pyruvic acid is converted into lactic acid in the absence of oxygen. Okay? The fate is decided now. If oxygen is available, the pyruvic acid completely oxidized into carbon dioxide and water. This is called aerobic respiration. If oxygen is not available, if the microorganism is the yeast, that pyruvic acid break down into ethyl alcohol plus carbon dioxide. As alcohol is formed, this is called alcoholic fermentation. And if it is bacteria, the pyruvic acid in the absence of oxygen is converted into lactic acid. And this is called lactic acid fermentation. This is called lactic acid fermentation. I told you one important point. The metabolic fate of pyruvic acid depends upon cellular need. If oxygen is available in the cells, the pyruvic acid fate is different. If oxygen is not available, the fate of pyruvic acid is different. Okay. Now we will discuss about next concept, that is fermentation. So we will see this one and this one. And in the next class, we go with aerobic respiration. Okay. So next heading, fermentation. What is meant by fermentation? Listen carefully. What is meant by fermentation? Fermentation is a metabolic process in which microorganisms partially oxidize glucose into pyruvic acid, and pyruvic acid is converted into either alcohol or acid, is called fermentation. I repeat, fermentation is a metabolic process in which glucose is partially oxidized into pyruvic acid, and this pyruvic acid is further converted into either alcohol or acid, is known as fermentation. And fermentation takes place under anaerobic conditions. It takes place under anaerobic conditions. It takes place under anaerobic conditions. It occurs in the cytoplasm. Fermentation takes place in the cytoplasm. It was discovered by Gay-Lussac. Right. Okay. Fermentation takes place under anaerobic conditions. It occurs in the cytoplasm, the same place where glycolysis takes place. Fermentation was first discovered by Gay-Lussac. Right? Now, the substrate for fermentation is pyruvic acid, because glucose converted into pyruvic acid. This pyruvic acid is the substrate for fermentation. It is converted either into alcohol or into acid. Example, lactic acid. Understand? Fermentation also occurs in the human cells when during heavy exercise, when oxygen concentration is very less. Fermentation also occurs in the human cells. Do you understand? Fermentation also takes place in the human cells during heavy exercise, oxygen is not adequate. In such a condition also fermentation takes place. Okay. And number of ATP formed in fermentation is 2. Remember, the ATP formed in glycolysis are 2. Those are also continue in the fermentation. Therefore, number of ATP formed in fermentation means you have to consider the 2 that is formed in glycolysis. So glycolysis is followed by fermentation if oxygen is not available. If oxygen is not available, glycolysis followed by fermentation. That means pyruvic acid enters into fermentation. So that means the ATP formed in glycolysis also considered in fermentation as well. Okay. So that means if a question is asked like that, how many ATP are formed in fermentation? 2. Actually, these 2 are formed in glycolysis. So those 2 are also there available in fermentation. Okay, right. And fermentation involve microorganisms. Microorganisms. Example, yeast, bacteria, right? And fermentation commonly found in prokaryotes. This is very, very important. Unicellular eukaryotes and germinating seeds. Germinating seeds. This is very, very important. Fermentation is commonly seen in most of the prokaryotes. Unicellular eukaryotes like yeast. Yeast is unicellular fungi. Fungi are eukaryotes. Bacteria are prokaryotes. And it also seen in germinating seeds. These are very important points. I will tell you once again. Fermentation takes place under anaerobic condition. The site of fermentation is cytoplasm. Okay. Fermentation was first discovered by Gay-Lussac, a scientist called Gay-Lussac. In fermentation, glucose is converted into pyruvic acid, and the pyruvic acid is finally converted into either alcohol or acid. For example, lactic acid, I told you. The number of net gain of ATP in fermentation is 2. Okay. And in fermentation process, microorganisms are involved. For example, yeast convert pyruvic acid into alcohol. Bacteria, that is lactobacillus, convert the pyruvic acid into lactic acid. I will show you later. And this fermentation commonly seen in most of the prokaryotes, unicellular eukaryotes, and germinating seeds. Right? Hope all of you are following. Okay? Do you understand, everyone? May I repeat once again? I am talking about what is fermentation. Everybody has to understand what is fermentation, where it occurs, why it occurs, when it occurs. You have to understand concept. is required. Conceptual learning is essential for success in competitive examinations. Don't by heart the subject. Concept is very important. Yes? Right. I hope you understand some points related to fermentation. Okay? Now we will see what are the steps involved in fermentation. First we will see alcohol fermentation, then lactic acid fermentation. Okay. First one, alcohol fermentation. Alcohol fermentation. Okay. You know already in glycolysis, glucose is converted into pyruvic acid. Glucose is converted into pyruvic acid, right? This is glycolysis. I need not say again, and this is glycolysis. Now, this pyruvic acid is converted into ethyl alcohol. Ethyl alcohol. It occurs in two steps. In the first step, this pyruvic acid undergo decarboxylation and form two molecules of acetaldehyde. Acetaldehyde. Here, undergo decarboxylation means carbon dioxide is liberated. This reaction is called decarboxylation, pyruvate decarboxylation. And the enzyme involved in this process is called pyruvate decarboxylase. This is the first step. See carefully, all of you. I told you fermentation occurs in two steps, correct? In the first step, pyruvic acid is converted into acetaldehyde. You know, glucose is a 6-carbon molecule. One 6-carbon molecule split into two 3-carbon molecules. Now each pyruvic acid undergo decarboxylation. Decarboxylation means removal of carbon dioxide. So each pyruvic acid undergo decarboxylation and one CO2 will come. So as two pyruvic acids are involved, two CO2. Therefore, 2 acetaldehyde. Acetaldehyde is a 2 carbon molecule. Clear? Acetaldehyde is a 2 carbon molecule. This reaction is called decarboxylation. This step is called decarboxylation. And the enzyme involved is called pyruvate decarboxylase. Pyruvate decarboxylase. Now, second step, last step. This acetaldehyde is reduced into the end product, that is ethyl alcohol. This is reduced into ethyl alcohol. See, 2 molecules of ethyl alcohol or ethanol, you can write. It is also 2 carbon molecule. Here, reducing agent is required. The reducing agent is NADH. This NADH formed in glycolysis is used in fermentation. Here key point is there. Very important point is there. Who is the reducing agent? Who is the reducing agent for conversion of acetaldehyde into alcohol? That is NADH. Where is formed NADH? In the glycolysis. So NADH formed during oxidation step of glycolysis is used for conversion of acetaldehyde into ethyl alcohol. Therefore, what is the net gain of NADH? What is the net gain of NADH in fermentation? Anybody? What is the net gain of NADH in fermentation? What is the net gain of NADH in fermentation? Nobody is giving the correct answer. Means you didn't understand. Or you will not concentrating. Only one person, Kirti again. Of course, thereafter many people are giving. Yes, later many are giving. Okay? Yeah, but some of them are also giving incorrect answers. Yes, 0 is the correct answer. Now everybody got it. Very good, very good. Everybody has to understand. We are not teaching for one person. Top to bottom, everybody should understand. Until you understand, we will teach repeatedly. That is our aim. Otherwise, what is the need of a teacher? Okay? So, the net gain of ATP will be 2, I told you. So net gain of NADH will be zero. Very good. 0. Why it is 0? Why it is 0? Because NADH is reduced. NAD is reduced into NADH in glycolysis. It is oxidized. NADH is oxidized into NAD plus. That means it acts as a reducing agent. Here simply you can say NADH formed in glycolysis, used in fermentation, hence net gain will be 0. Net gain will be 0. This step is called reduction. This step is called reduction. This step is known as reduction step. And the enzyme involved in this step is known as alcohol dehydrogenase. Alcohol dehydrogenase. Alcohol dehydrogenase. Right? This is alcohol fermentation. This is called alcohol fermentation. See all of you. Right now I will ask few questions on this. Few questions. End product of alcohol fermentation. End product of alcohol fermentation. Why dragon is angry? Why dragon is angry? End product of alcohol fermentation is which alcohol? Ethyl alcohol. Ethyl alcohol. Yes. Very good. Ethyl alcohol. Second question. How many steps are involved in alcohol fermentation? How many steps are involved in alcohol fermentation? Very good, very good. 2 steps. But very few students are giving 4 steps. Only 2 steps in alcohol fermentation. Yes. Can you say what are those 2 steps? Can you say what are those 2 steps? Names of the steps of alcohol fermentation. See, decarboxylation first step, reduction second step. Decarboxylation first step, reduction second step. Okay, next question. How many enzymes are involved in alcohol fermentation? How many enzymes are involved in alcohol fermentation? Again 2, very good. Can you say what are those 2? Can you say what are those 2? See, very clear. Pyruvate decarboxylase, first enzyme. Pyruvate decarboxylase, first enzyme. Alcohol dehydrogenase, second enzyme. Correct? Pyruvate decarboxylase, first enzyme. Alcohol dehydrogenase, second enzyme. 2, right. 2 more last questions. Net gain of ATP in fermentation. Net gain of ATP in alcohol fermentation. Net gain of ATP in alcohol fermentation. Not 0, 2. Very good, 2. Last question. Net gain of NADH in fermentation. Net gain of NADH in fermentation. 0. 0. Very good. Right? You know, one important point. In anaerobic respiration, the energy present in the glucose liberated only less than 7%. Because only 2 ATP is liberated, right? In fermentation or in alcohol, in anaerobic respiration. In anaerobic respiration, the energy present in the glucose, total energy present in the bonds of the glucose, how much energy is liberated? Only 7%. Less than 7%. Understand? And moreover, accumulation of this alcohol in the cells where fermentation is going on is hazardous, means dangerous. Accumulation of alcohol or acid in the cells is hazardous to the cells. And finally, the yeast cells will die during fermentation when the alcohol reaches to 13%. Understand? When alcohol reaches to 12 to 13%, the yeast cells will die. Now you have one doubt. There are many alcoholic beverages contain 30% alcohol, 40% alcohol, but yeast cannot tolerate more than 13%, 13%. Then how high percentage of alcohol containing beverages are made? Do you understand my question? In this alcohol fermentation, the yeast can survive up to 13%. Understand? There are many beverages which contain more than 30%. Some may are having 30% alcohol, some may are having 40% alcohol, some may have even greater than this also. Then how these beverages are made? Only the yeast can tolerate, because this alcoholic fermentation is caused by yeast. That is Saccharomyces cerevisiae. This is the name of the common yeast. Saccharomyces cerevisiae. Scientific name of yeast. Scientific name of yeast that is commonly used in alcohol fermentation is Saccharomyces cerevisiae. This yeast, during this fermentation, when the alcohol reaches to 13%, the yeast will die. My question is, by what process the beverages containing more alcohol greater than 13% are made? By a process called distillation. Okay? So by using distillation process, beverages having high percentage of alcohol are made. So remember, this is about alcohol fermentation. And lastly, the next one is lactic acid fermentation. Lactic acid fermentation. In lactic acid fermentation, see all of you, first glucose break down into pyruvic acid, same pyruvic acid. This is 6 carbon compound, this is 3 carbon molecule. As a result, 2 molecules are formed. This process is called as glycolysis. Right. Now, this pyruvic acid enters into fermentation. This is called lactic acid fermentation. In this fermentation, lactic acid bacteria are involved. Okay. In this process, the pyruvic acid is converted into lactic acid. Lactic acid is a 3 carbon compound. Okay. Here the type of microorganism is changed. Therefore, the type of product also changed. The type of product produced depends upon the type of microbe used. If the microbe is the yeast where ethyl alcohol is produced, if the microbe is the lactic acid bacteria, lactic acid is formed. That is called alcohol fermentation. This is called lactic acid fermentation. Okay. Pyruvic acid is converted into lactic acid. In this process also, the reducing agent is same, NADH. NADH converted into NAD plus, same. And the enzyme involved in this process is called lactate dehydrogenase. Lactate dehydrogenase. This is the enzyme that is involved in lactic acid fermentation. Okay. So, with the help of lactic acid fermentation, with the help of lactate dehydrogenase enzyme, pyruvic acid is converted into lactic acid. An important point is CO2 is not liberated. CO2 is not liberated in lactic acid fermentation. Directly pyruvic acid is converted into lactic acid with the help of an enzyme called lactate dehydrogenase, and this is called as lactic acid fermentation. In this, the microorganism involved is lactic acid bacteria. Lactic acid bacteria are involved. Understand? Lactic acid bacteria are involved. Example, lactobacillus. This is called lactic acid fermentation. So in lactic acid fermentation, finally I am concluding, glucose split into pyruvic acid. This whole process is called glycolysis. Now this pyruvic acid in the lactic acid bacteria directly converted into lactic acid with the help of an enzyme called lactate dehydrogenase. The reducing agent involved is NADH formed in glycolysis. Therefore, the net gain of NADH will be 0 here also. Number of ATP formed in lactic acid fermentation are 2. Net gain of ATP in lactic acid fermentation 2. Net gain of NADH will be 0. And the enzyme, only one enzyme is involved. That enzyme is called lactate dehydrogenase. The product of lactic acid fermentation is lactic acid. So that's all, students. So this is about lactic acid fermentation. So with this, fermentation is completed. Both alcohol fermentation and lactic acid fermentation. And that's all today. Hope you understand, everyone. And we will continue in the next class. Thank you.